All right, welcome to your first lecture on bioenergetics. Over the past several years, I've noticed that certain lectures are a little bit more difficult to comprehend, a little bit more tedious, and so it's hard to pay attention in class and take notes. So, what we're doing this semester is those difficult lectures we're going to put online for you to be able to go over it over and over again, and then we'll bring you into class and we'll discuss in person then. So let's get into it. First off, my first question to you is, what is bioenergetics? Well, like the name suggests, it's essentially the control, the processes of energetics in the human system, or the flow of energy in biological systems. We could also refer to it as the conversion of macronutrients into biological useful forms of energy. So what's we got here? To your left, carbohydrates. In the middle, we have butter. To the right, you have steak or what? Protein, right? What is our biologically useful form of energy, or the universal source of energy currency that we use? Well, as you know, it's ATP, or adenosine triphosphate. So ATP represents the energy currency that powers all forms of biological work. We just like to think of muscle contraction, but nerve transmission requires work, secretion of hormones, digestive system. If you take amino acids, for example, and form a protein, that requires ATP, and even circulation. So almost everything that we can think of requires ATP. So how does it work? Well, one of the things to know is that ATP, or adenosine triphosphate, is an adenosine molecule bound to 3 high-energy phosphate groups. So these phosphate groups are bound by high-energy bonds. So in a real simplistic term, you kind of want to think of it like a dart gun. You load the dart into the gun, and that takes energy. But when you pull the trigger, that energy is released and forms kinetic energy, or the energy of movement. Same thing with the ATP molecule. If we break off a phosphate group, we can actually harness that energy that was stored in the bond to power work. You can also take the ADP molecule, break another phosphate group off, and use that energy. But primarily in this class, we're more going to be talking about ATP being converted to ADP. Now, there's 2 processes that I want you to be cognizant of, and that is anabolism, which is one of my favorite terms, and catabolism, or catabolism. So anabolism essentially is the building of larger molecules from smaller building blocks, or smaller precursors, right? So if you take several amino acids and you form a protein, that's important. That requires energy. But if you break down molecules, for example, when we break down carbs, fats, and proteins, that releases energy, and that's a catabolic process. So anabolic requires energy, catabolic releases energy. It's also important to understand that these processes are typically coupled, right? So if you're breaking down ATP, that's a catabolic process, but if you're using it to build up proteins, that's an anabolic process. So we have 3 basic energy systems that exist in our muscle cells to replenish ATP. These include the short-term phosphagen system, which is our most explosive system, the glycolytic or glycolysis, which uses carbohydrates, and the oxidative system, which, as the name suggests, requires oxygen. If you look at these energy systems, the phosphagen system has about 72 kilocalories, and it has a capacity of 0 to 10 seconds, while glycolysis uses about 36 kilocalories a minute, but only lasts from 30 to 90 seconds. Whereas oxidative has a power of 18 kilocalories a minute, but could last with almost infinitely, right? If you're just using carbs, 2.5 hours. If you're using purely fat, it could be almost infinite, because we have like 40,000 calories worth of fat or more. So, what I want you to think about is this. The more powerful the system, for example, phosphagen, the quicker or the lower its capacity. So it's only 10-second capacity, but 72 kilocalories a minute. You cut that in half for glycolysis for 36 kilocalories a minute, but it's 30 to 90 seconds. You cut that in half for oxidative, but it's almost infinite, right? So the more powerful the system, the lower its capacity. So if we talk about the phosphagen system, it's the most powerful. It lasts about 1 to 2 seconds of work. So if you actually, that's ATP. ATP is about 1 to 2 seconds of work, but you have 4 to 5 times more phosphocreatine in the body. And phosphocreatine can be used to replenish ADP to ATP. So think about it. You have ATP. You break off a phosphate group. Now you have ADP plus a free phosphate and energy. Well, creatine phosphate, if you look at the graph below, donates its phosphate group to ADP. Now you have free creatine and ATP. So that whole process lasts about 10 seconds. Because it is such a rapid system and so powerful, it essentially will last us for movements like vertical jump, swinging a bat, throwing a punch. If I told you to stand up right now as fast as you could, you'd be using the phosphagen system. So one of the questions that I always get is, is that the same thing as like what happens when you supplement with creatine? The answer is yes. Okay, but some people will say, if you supplement with creatine, that works by drawing water into the muscle, and that makes the muscle get bigger, right? That's the layman theory, but when you stop taking it, your muscle deflates. Yeah, that theory's false. Creatine supplementation, what it does is it saturates the creatine phosphate system. So now you have more creatine phosphate, and now, for example, instead of getting 8 reps, maybe you get 9 or 10. If you've gotten 9 or 10 instead of 8 or 9 reps or 8 reps, then you're going to have a greater training stimulus, and therefore you're going to get greater growth. So this is a study by Volek, a friend of mine. If you look at the creatine condition, you get greater increases in strength in the creatine condition and the bench press condition. And that's the striped condition, when you take creatine versus not taking it. And then if you look at type 1, 2A, 2AB, and 2B fibers, these are basically your slow and different variants of fast twitch fibers. See, the creatine group has larger size on all accounts. Moving on to glycolysis. Glycolysis has 2 possible end products. If you're going slow, like the lady running, then the end product that you're going to be using is going to be pyruvate. So you start with glucose, which is a 6-carbon molecule, and you break it down essentially into 2 pyruvates. In the process of breaking down glucose to pyruvate, you form 2 energetically useful products. The first is NADH, 2 of them, and then ATP, 2 of them. So you form 2 ATPs and 2 NADHs. Now, pyruvate can then enter into the mitochondria and be used via the oxidative system. However, if you go very high intensity, like 30 to 60 seconds all out, like if you're a hockey player in a shift, then the byproduct, what happens is that you're producing so much pyruvate that your mitochondria can't keep pace. And so to keep glycolysis running, pyruvate gets converted to lactic acid. And that's when you get that acidic buildup in the muscle. Okay, so 2 different sort of pathways. But still you're forming 2 NADHs and 2 ATPs. And again, this is what I want you to know. You form ATP, NADH, and pyruvate or lactate. So how do we deal with lactate? We know when we have lactic acid, we have acidity, but how do we clear it? How do we deal with it? Well, two ways. Lactate can be transported in the blood to the liver, where it's converted to glucose, and that's named the Cori cycle. So basically, lactate's formed in the muscle. It's transferred in the blood to the liver. The liver converts it to glucose. That goes back to the muscle. Gets converted to lactate. That goes back to the liver, gets converted to glucose, goes back to the muscle. You get the point. And Cori discovered this cycle, and so it's called the Cori cycle. Lactate, on the other hand, can be transferred to the heart and used as fuel as well. The heart loves lactate. Remember, it's still a very energetically useful molecule. So how do we deal with lactate? Well, one of the things we can talk about is active versus passive recovery. Think about when you're training, right? If you see a guy and they do squats, they'll go heavy on squats, and then what do? They sit down on the bench and they recover. That's called passive recovery. Then you might have a girl might do squats and she wants to keep her heart rate up, so she paces around the gym. That's called active recovery. Okay? Now, there are a couple ways to do active recovery. One would be just to lightly pace around the gym, but also sometimes you see trainers put people on the treadmill and have them jog. That would be active, moderate to hard active recovery. Okay? So in this study, what they did was they looked at blood lactate levels, and they had people do 4 sets of 10 repetitions, and you look at blood lactate with passive recovery versus active light and versus active moderate. Which condition do you think resulted in the least amount of lactate? I'll let you think about that for a second and tell me why would that be the case. So which condition and why? All right, you have your answer? All right, let's look and see if you are right. This is passive recovery, sitting down. That's active light, and that's active moderate. So why is this the case? What's going on here? Well, first off, active recovery, what's it doing? If you're sitting down, for passive recovery, you're sitting down, the lactate's just pulling in your muscle, and it can't be circulated to the liver or the heart. But if you do active recovery, you're pumping the lactate to the heart and to the liver, which is helping you clear it. So then why moderate? Why is moderate no better than passive? Because even though you're pumping blood fast, you're producing lactate at the same rate that you're clearing it. So this is why your active recovery should be a very light activity like walking. And then let's look at who got the most repetitions. This is the passive condition. That's the moderate condition, active moderate. That's the active light, and that's the active moderate. So notice the active light, which is the green, got the most reps. So theoretically speaking, they should also get the greatest gains, at least in strength. So this is very important for your athletes. And we see this in football, for example. After a down, or you switch from offense to defense, then you may have football players, for example, lightly riding the stationary bike. Okay, let's move on to oxidative metabolism. Remember, during high-intensity exercise or low-intensity exercise, glycolysis converts to pyruvate. And pyruvate ends out going to the mitochondria, where it is converted to acetyl-CoA. So, acetyl-CoA essentially is converted into something known as citric acid. Okay, so let's take a look at what this looks like. Okay, we have pyruvate. Okay, and we form 2 pyruvates per glycolysis, right? Pyruvate gets converted to acetyl-CoA, and that gets converted into citric acid, right? So you start with citric acid, and essentially that gets catabolized or broken down. During that process, you form NADHs, you form FADHs, and you form ATP. Now, ATP, you know why that's useful. NADH, you remember you formed also in glycolysis, but FADH I haven't talked about. So remember NADH and FADH. Those are very, very important molecules, and in fact, we produce the most energy from these. Again, I'm pointing at NADH, FADH, and ATP. So what do NADH and FADH do? Well, they carry electrons. One of my old professors when I was an undergraduate, Dr. Kathy Inouye, said, Jacob, you kind of want to think about NADH and FADH as taxi cab drivers for electrons. Now, the analogy was kind of simple, and, I shook it off, but today I look back and go, oh, it's kind of a good analogy. So, if you look at NADH and FADH, they're very energetically important. And you'll understand what they do when you talk about the electron transport chain. And the electron transport chain is found in the mitochondria, particularly in the inner membrane, right? So you have this inner membrane. And in the inner membrane, you have what we call electron carriers. Now, NADH and FADH come down to the electron carriers and they drop off their electrons to the first electron transport protein. Now, each electron transport protein in this chain has a stronger attraction for electrons than the previous one. Okay, so let me say this again. NADH and FADH drop off their electrons to their electron transport carrier. The next electron carrier in line has a stronger attraction for electrons. So where are the electrons going to go? To the next electron carrier, and the next, and the next. Okay, so basically it's almost like if you took a boulder and pushed it downhill. It's just going to go, 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 go. So the electrons are falling down the chain, and that produces energy. That energy is used to power hydrogen pumps, or hydrogen ion pumps. So you have hydrogen ions in the inside of the mitochondria, which is known as a matrix. The proton pumps, or hydrogen pumps, use the energy from falling electrons to pump hydrogens outside of the mitochondrial matrix into what we call the inner membrane space. I'm pointing to that with that red arrow. Now think about this for a second. If I'm using energy to pump hydrogen ions to the inner membrane space, there's now more hydrogen ions in the inner membrane space than the matrix, or the center of the mitochondria. Okay? As you know, things are going to diffuse to reach an equilibrium. So if I pumped hydrogen ions in the inner membrane space, where do they want to go now? They're going to want to flow back into the matrix. Okay? And what happens is that they flow back into the matrix through an enzyme called ATP synthase. Do you see that to the upper right? So they're going to flow back through the enzyme ATP synthase. What does that enzyme do? Well, think about the name, ATP synthase. It synthesizes ATP from ADP. Okay, so going back again, we formed NADH as well as NADH and FADH in the glycolysis, and then both of them in the Krebs cycle. They brought their electrons over the chain, dropped the electrons to the first transport protein. That moved to the next one, had a stronger attraction, and flew down the chain. Proton pumps, or hydrogen ion pumps, were used to pump hydrogen ions into the inner membrane space, creating a concentration gradient. Those then flew back into the matrix, and the flowing back through the ATP synthase enzyme triggered the enzyme to form ATP. This is an overview. NADH and FADH bring electrons and drop them off at the transport chain. Electrons are moved down electron carrier proteins, each with a stronger affinity for the electrons in the previous complex. The energy from falling electrons is used to eventually power the ATP synthase enzyme, which rephosphorylates ADP back to ATP. One of the things I want to point out is the concept of specificity. Okay? Think about this for a second. What is specificity? First off, can you say that term, specificity? I hear some people say specificity, right? So it's a little tricky term, but what is it? Well, I think everyone can kind of define it, but I find that 90% of the people, even though they define it, don't understand it. Okay? Specificity means this, that if you want to acquire a specific outcome, for example, dunking a ball, okay, or swinging a bat, that your training should mimic that as much as possible. Now, people will often misinterpret that, and I'm going to get on my high horse here, okay? As you'll see, I'll do a lot in class. But you'll see, for example, a strength coach will say, oh, this football player is going out of bounds when they catch the ball, before they catch the ball. It must mean that they need balance. So what they'll do is they'll put them on a Botsu ball. Well, unless you're playing football and there's an earthquake, that's probably not going to transfer to balance on the field. In reality, that football player needs to practice catching the ball without going out of bounds over and over again in practice, right near the out of bounds boundary. Okay, so it's very, very important about specificity. When you see a strength coach and they have a workout on the board and every team does that same workout, they are not using specificity. One of the things is our strength coach, Coach Justin, is outstanding at using specificity. You know, everything he does, he's programming based on the sport. But our previous strength coaches before Coach Justin weren't as good at specificity. I saw them taking our baseball players and having them do circuit training. Why? How does that mimic the sport? It doesn't. So the first level of specificity is metabolic specificity. You should train with an intensity and duration that stresses the primary energy system used for the event. Okay? So if you're a power lifter, you should stress what system? ATP PC. If you're a long distance runner, what system should you stress? Oxidative, right? So that's what I want you to think of when you're thinking about metabolic specificity. Last thing I'm going to talk about today is excess post-exercise oxygen consumption. Exercise itself has an impact on energetics. And oxygen requirements are a reflection of how much energy we're expending. So when we're done exercising, our oxygen demands go up because our work demands go up. Okay, so this is exercise. Basically, we exercise, oxygen goes up, and then it hits a steady state. So say that I were to run 6 miles an hour, which doesn't happen very often. But if I say I were to run, oxygen would go up, and then it would plateau. Now, if I stop training, oxygen is going to rapidly go down, and then it's going to plateau out. If you were to look at that, can you distinctly see two phases? Yeah. If you look at the first phase here, that's a rapid phase. Okay, we also call it the alactic acid phase. Then we have a slow phase, or the lactic acid phase. So this rapid phase is basically characterized by a number of things. One, it's not used to convert lactic acid back to glycogen. It's mainly used to resynthesize ATP and creatine phosphate. It's also used to replenish myoglobin with oxygen. Now, first off, where do we carry oxygen in blood? It's in the red blood cells, right? On what molecule? Hemoglobin, right? So if you look in the muscle, we carry a single oxygen on a myoglobin molecule. So we're replenishing myoglobin and we're replenishing ATP and creatine phosphate. The slow phase is everything else. Reconverting lactic acid back to glucose and then glycogen. When you're done exercising, your temperature's elevated. That explains most of the reason why your metabolism's elevated. Hormones are still elevated. Your heart's still beating faster. When you damage your muscles, they have to be repaired. So that's protein synthesis. All of these things result in elevated metabolism for as much as 24 hours or more. So do you think that elevating your metabolism after exercise could be good for weight loss? Absolutely. So let's look at what affects metabolism. What are the two things during exercise that might affect metabolism? Think about that for a second. I'll let you think about that. All right, what's your answer? Well, this is looking at what I'm talking about here. So we're looking at is exercise intensity. Notice that, say this percentage of your heart rate max. This is looking at excess post-exercise oxygen consumption, which is what I'm telling you about. So elevated metabolism after exercise is reflected by excess oxygen consumption. So we call that excess post-exercise oxygen consumption. So if you look at this, what we can see is that you don't increase your metabolism or excess post-exercise oxygen consumption till about 50-60% of your heart rate max. And then it goes up exponentially, right? So it's 50 to 60% threshold. Then you go up drastically with every increase in intensity. Also, if you look at duration, the longer you go, the greater the excess post-exercise oxygen consumption. So the problem, though, is if you go long duration, you can't go high intensity, right? You're going to peter out. So one study looked is, can you combine the two? And how would you combine the two? How could you go high intensity and long? Well, what would that be called? What training method? All right, what's your answer? Well, it's interval training. So what you're looking at here is they did one group where they trained at 60% of their heart rate max for the whole exercise session. And the other group did an average of 60 by alternating between 30 and 90% of their heart rate max. But on average, they had 60%. Who do you think had greater excess post-exercise oxygen consumption? They all had the same average 60%. The answer, as you might suspect, was the interval group. Here, we see the moderate group, that's how much oxygen they were consuming more, and that's the interval group. Okay, so that's the end of the first lecture. I'm going to put a series of questions up on your quiz, and they'll be all related to the lecture. And I'm also going to put, you'll also see on Blackboard, you have a little guide of questions that you should be able to answer for this lecture. So on Tuesday, we're going to come into class, and you're going to be prepared to answer all these questions because we're going to literally take the lecture just to review the material of this. Because again, it's a hard lecture, and I want to make sure you guys have the material down. Thanks for listening to the lecture, and I'll see you guys in class.